In this final video of Unit 2, we are going to see in detail about the different types of RNA and its crucial role in the synthesis of protein. So already we know RNA is a single-stranded structure and it has 4 bases: adenine, guanine, cytosine, and instead of thymine we have uracil. So this RNA are classified into 4 types: mRNA, tRNA, rRNA, and miRNA. So in this particular video, we are going to concentrate on the first 3 types of RNA, which is very crucial for the production of protein. So the first type of RNA is called as mRNA, or in brief, messenger RNA. This messenger RNA plays an important role in the transcription of DNA. So what is transcription? So transcription is the process in which the protein synthesis that involves copying the genetic information contained within the DNA into a RNA message. So basically what happens here is the DNA stays inside the nucleus. So it can't take out the message out of the nucleus by itself. So it needs a helping hand. That's where your mRNA comes. So the DNA transfers or transcribes the message into a form called as messenger RNA. The DNA contains the 4 nucleotide base A, T, G, and C, which are paired together in the form of A to T and C with G. When RNA polymerase transcribes the DNA into a mRNA molecule, the adenine pairs with uracil, which is the change happening here, and the cytosine pairs with guanine. At the end of the transcription process, the messenger RNA is transported to the cytoplasm from nucleus, where it's going to complete the process of protein synthesis. You could see here, this is an image of a pre-mRNA and becoming a mature mRNA. So initially, after the transcription process, you will have something called as pre-mRNA. So it has few regions called as introns, and there are few regions called as exons. So introns are some unwanted regions or non-coding regions. That means it's not going to code for protein. And exons are the region which is going to code for your proteins. So this pre-mRNA undergoes some processing and these introns are removed out and all these exons have joined together and become a mature RNA. Once this mature RNA is ready, it leaves away the nucleus through the nuclear pores and reaches the cytoplasm, where it gets ready to synthesize the protein. The second type of RNA is called as rRNA, that is ribosomal RNA. This ribosomal RNA is a component of cell organelles, which is called as ribosomes. So a ribosome consists of ribosomal proteins as well as rRNA. So it's not just made up of rRNA, there is a combination of few ribosomal proteins also. These ribosomes are typically composed of 2 subunits. You could see in the image here. Okay, so this is the 2 subunits. One is called as a larger subunit and the second one is called as a smaller subunit. So this larger subunit and smaller subunit together form your ribosomes. These ribosomal subunits are synthesized in the nucleolus by the nucleolus. So already we have seen in Unit 1, inside the nucleus we have an organelle called as nucleolus. This nucleolus is responsible for the synthesis of these 2 ribosomal subunits. Ribosomes contains a binding site for the mRNA. So in previous slide we have seen about mRNA. So once the mRNA gets matured and leaves the nucleus, it reaches the cytoplasm where it meets with the next type of RNA, that is rRNA. So in this rRNA, it has sites. We can see in this image, P site, A site. Actually, there will be like one more site, it's called as E site. So we can remember easily by the short form APE. Okay, so you would have seen few movies related with A. Try to correlate with. So A, P, E are the 3 sites where your mRNA is going to come and bind. So ribosomes contain a binding site for mRNA and 2 binding sites for tRNA located in the larger ribosomal subunit. So during the translation process, which is the next process, okay, so initially you have a transcription which is followed by translation. So during a translation, a small ribosomal subunit attaches to a mRNA molecule. So you could see here there is a small subunit and it gets attached with the mRNA which reaches the cytoplasm. At the same time, an initiator tRNA, you could see here this is initiator tRNA. This particular molecule recognizes and binds to a specific codon sequence on the same mRNA molecule. So what is a codon? So codon is nothing but a triplet of a nucleotide. For example, A, T, G. So 3 codon, if it's there, it's called as a codon. It's a triplet. Okay. So ribosomal RNA is responsible for creating the peptide bonds between the amino acids in the polypeptide chain. So during the synthesis of protein, already we have studied proteins are nothing but a chain of amino acids. This chain is made by the bond called as peptide bond. This ribosomal RNA is the one responsible for creating this peptide bond between these amino acids and finally giving us the polypeptide chain. We call it as proteins. The last type of RNA is called as tRNA. So we call it as transfer RNA because it's going to transfer amino acids to help in the synthesis of proteins. This transfer RNA plays an important role in the translation portion of protein synthesis. Its job is to translate the message within the nucleotide sequence of mRNA into specific amino acid sequence. Already we have seen there's a DNA which is transcribed its message into a mRNA. Then mRNA reached the cytoplasm. Now this transcribed message has to be translated again to form a protein. So that's where your tRNA plays a major role. The amino acid sequence are joined together to form a protein. The transfer RNA is shaped like a clover leaf. So this is the shape of a tRNA which looks like a clover leaf with 3 hairpin loops. It contains an amino acid attachment site on one end. So you could see here there are 3 loops in your tRNA shape and it has an amino acid attaching site. Okay, it is called as the anticodon site. So in this anticodon site, it has already we have seen what is a codon in our previous image. So codon means 3 nucleotide base pairs. So this anticodon is going to be complementary to your codon. So for example, if it's in your mRNA, if it's like CGG, your anticodon will be like this GCC, which is like complementary to your or pairing with your mRNA strand. This anticodon recognizes a specific area on mRNA called a codon. So already as I said, a codon consists of 3 continuous nucleotide bases that codes for an amino acid or signal the end of translation. The transfer RNA along with ribosomes reach the mRNA codons and produce your final polypeptide chain. So this is how a protein is synthesized. This is where these 3 types of RNA plays a very important role. So with that we come to the end of Unit 2 Biomolecules.